Hi, everybody. I'm Jenny LeRae, and I want to welcome you to today's Research America Alliance discussion. Thank you for joining on Zoom and for your partnership in the Research America Alliance. We always start off with some housekeeping. If your organization is not a member, please reach out to Kristen Furlong, whose email is in the chat. She's our Senior Director of Membership and Development, and she can tell you more about our wonderful work and our alliance. So please type your questions into the Q&A box or the chat box, and we'll get to as many as we can during the audience Q&A portion of today's session. Today, we're having an interactive discussion. Some folks call it a fireside chat with the several members of the All of Us research program. And we're going to really focus on the All of Us research program's efforts around chronic disease, particularly diabetes and other high burden chronic conditions. Across the nation, volunteers have helped power research through All of Us that is breaking new ground against some of our nation's biggest health threats, and we're going to learn more about how they're doing that today. So it's a pleasure to introduce our three guest speakers, Dr. Josh Denny, who's the CEO of the All of Us research program at NIH, Dr. Holly Nicastro, who's the coordinator of the Nutrition for Precision Health program at the Office of Nutrition Research at NIH, and Dr. Rick Woychik, Director of the National Institute of Environmental Health Sciences and the National Toxicology program at NIH. Welcome to all of you. Thank you so much for joining us. We're going to start with you, Josh. Dr. Denny, it's great to have you with us. For those who aren't familiar, can you give us the Cliff Notes version of the All of Us research program and why it's unique?
Jenny, well, first, thank you so much for hosting this and Research America for pulling us together. And it's a real pleasure to be here with my colleagues, Dr. Nicastro and Dr. Woychik, to talk about All of Us and what we are doing here together across the NIH. First, the All of Us is really the U.S. national cohort designed to tackle vexing health problems across the intersection of lifestyle, biology, environmental exposures, nutrition, through a completely national cohort. We have all 50 states represented. Over 860,000 participants have joined us. And they've done things not just like in traditional cohorts where they might share survey information, go through a physical exam, maybe biospecimens, but we've also collected electronic health records across the myriad of different health systems we have across the country, combining those data together, harmonizing and breaking down some of those silos that we know exist in our healthcare industry. We have involved and created a platform that these participants can share digital health technologies like Fitbit data. They share their biospecimens include genomics. They can get genomic data back as well as we generate whole genome sequences, what will be the largest whole genome sequence data set in the world for research next year. We share these data back to participants. And then importantly, we're a platform to let other people do research. And that's really what we're talking together today about, is how All of Us helps researchers do work, helps propel research across the NIH. It's not just our study team. In fact, it's generally not our study team. It's 18,000 researchers that are doing the work that are not part of the All of Us program, that are building projects on top of it, making new discoveries. And Rick and Holly will really talk about two examples of this today.
Great. Thank you, Josh. Before we go into more specifics, can you share how you see All of Us driving progress towards national health priorities, particularly around chronic disease? I know we're going to get into more details with Rick and Holly, and also targeted treatments.
Definitely. We are a disease agnostic cohort. That means people enroll from across the country. We know they're a little bit sicker than the average population, but they basically represent all the communities across the United States. And so that means we have tens of thousands of people with, chronic disease, in some cases, probably exceeding 100,000 with common things like hypertension. We collect all age ranges, so we can really follow health across the lifespan. And we were working on even expanding to include children as well as part of families to help tackle some of those issues as well. So chronic disease, well represented in our cohort. We think about chronic disease as composites of genetic influence, environmental exposures, lifestyle habits. We're able to look at that intersectionality in the program, to look at things like nutrition. And that's one of the topics we're going to be talking about today. And, the risk of having multiple chronic diseases and what the outcomes of those would be, how different medications will work. How can we target hypertension therapy better? How can we tackle obesity? And what's the impact of tackling obesity with like GLP-1 drugs? What is the impact of those kinds of outcomes over time are things that we can really help address and look at the genetic stories to discover new treatments. The latest treatments we have for cholesterol, which are incredibly powerful, come from genetics. And we think we can continue to uncover those kinds of stories that will drive precision therapy for chronic disease in ways that are, radically transparent and reproducible as well, because everyone can come to the same data set and look at what's going on there.
Thank you, Josh. Well, that's a great segue into Holly. Can you share, Holly, how the Nutrition for Precision Health program is generating data and other information that will improve, the well-being of individuals and families?
Yes, happy to. And thanks for having me here. Thanks for the opportunity to talk about such an exciting study. So Nutrition for Precision Health, or NPH, it's a bit of a mouthful. We launched it so that we can move beyond one size fits all for diet and nutrition. We know that people respond differently to different ways of eating, and we're looking to uncover why. What are those factors that predict why two people eating the exact same foods could have opposite biological responses? If we can predict, we can tailor guidance to help optimize the health. So unlike traditional nutrition trials, we're not looking to see which dietary pattern or which way of eating wins. We're looking to see who wins on each way of eating. So we've embedded this study in the All of Us research program. All of Us research program, hundreds of thousands of individuals. NPH is a deeper dive study on at least 8,000 of those individuals. And we're collecting a breadth of data, new data that hasn't been previously collected in All of Us, things like the microbiome, additional wearables like accelerometers and continuous glucose monitors. And we're leveraging the very engaged All of Us participant cohort and the infrastructure and economies that the program provides.
And you said it was 8,000? Is that what you said?
Yes, 8,000 individuals.
Okay, that's great. And do you do interviews, surveys? What are some of the methods that you use in terms of collecting the information?
We're using a combination of observational approaches and different dietary intervention tests. So for the 8,000 individuals, we're following them for 10 days at a time. We're using new technologies to collect information about what they're eating. So we're using traditional methods, including dietary surveys and asking people what they ate, but we're also using tools that allow people to either take pictures of the food with their cell phones or to capture pictures of the food with tiny cameras that attach to somebody's eyeglasses. They're activated by chewing and then take pictures of the food. So it really eliminates the memory issue of having to remember what you ate throughout the day. We can get that objective data. But we're collecting surveys. The participants answer for us, many clinical measures, and everything is tied together by some standardized meal tests that we're providing to the different participants to gather a phenotype on them, on how they respond to different meals.
So these are longitudinal studies. It's not a cohort study. It's not a study where you have a control group.
Right. This is a short-term study. Most people, all of the 8,000 will do the 10-day observational period, and we'll have smaller subsets where participants will receive prescribed diets in different orders. Everybody will get all 3 diets. So there's no control arm compared to a different diet. It's how does each individual respond on each of these 3 different diets? And we're doing that in 2 different ways. We have one part where participants receive the food to take home and eat throughout their daily lives. And we have a smaller subset of participants who will actually check into the study center for 2 weeks at a time and live there and eat all of their food under direct observation.
I'm glad I'm not one of those people checking in, because I'd have to have someone sneak me a lot of chocolate. When will you have the first tranche of data?
We expect to have the data set ready to release publicly on the All of Us Researcher Workbench around 2027.
Okay, great. Or we're going to turn to you, Dr. Woychik, Rick, and we're going to learn more about your study, the NIEHS supported study connecting environmental exposures with chronic illness and long-term health concerns.
Great.
So Rick, share information about your
Well,
study.
Josh indicated in his introductory comments, I mean, we're very interested in the same chronic disease phenotypes and conditions that Josh is interested in from a genetic point of view. But as he indicated, that things are really gene by environment effects. So in most cases, it's not just your genes and it's not just your environment. And we know the environment is important because there are, I mean, countless examples of environmental exposures that lead to some of those same disease phenotypes that Josh is studying from a genetics point of view. I mean, I think we all know about lead and decreased IQ in children, behavioral problems, ADHD, arsenic, as it relates to predisposition to cancer, PFAS, these perfluorinated alkyl substances that seem to be ubiquitous in the environment. These are these forever chemicals. They actually cause immunotoxicity, liver injury and dysfunction, alter metabolisms, flame retardants, pesticides. In fact, there's an example of a pesticide, rotenone. If you want to create a Parkinson's rat or a mouse, treat it with rotenone, this pesticide. And of course, air pollution. I mean, particulate matter, PM 2.5. You can just look at the list of things that we know are associated with air pollution. There are respiratory, cardiovascular, neurological, immune, metabolic, and neonatal functions. Now, prior to joining up with Josh and the All of Us program, the environmental health sciences community was doing its thing with environmental health, with the environmental exposures, and Josh was doing his things genetically. But the real solution to get to the core understanding of chronic disease, we got to do both. We got to do genetics and we got to do environment. And that's where it's really Josh has been just a fabulous partner working together with us. Now, we also know that individuals respond, that, Holly was talking about how individuals respond to diet differently. They respond to the environment in different ways. And so we have many examples where there are cases where we know there are certain genes, if there are mutations in these genes, that increases the risk to Parkinson's disease or Alzheimer's or other things like that. But most of these are complex traits, the things that Josh and the All of Us program is really built to study. The genetic, the heritability that involves, tens, if not hundreds of different gene variants that all contribute to this heritability. So the best way to do this is to partner with this fabulous cohort. So I'm a big supporter of what Josh does, and I'm a big supporter of the All of Us program, and we need to be moving forward on this. Last comment that I'll make is that just like the genome project, went beyond one gene at a time. If we want to understand the genome, we have to go beyond one gene at a time. The environmental health sciences community has come to recognize that if we want to understand the influence of the environment, we have to go beyond one exposure at a time. There can be synergistic effects between different physical chemical exposures, but we absolutely must include nutrition, exercise, sleep, and other social determinants that relate to the environmental exposures. So we're advancing this new concept called the exposome. Multiple exposures involving multiple physical, chemical, as well as social determinants, and really developing a strategy, a framework, and how we can bring the exposome to Dr. Josh Denny and his colleagues that are part of the All of Us program. And in the end, really bringing together a gene by environment framework.
Wow, that's very interesting and ambitious, as it should be, coming from NIH. Rick, when do you think you'll have some early findings?
Well, we already have a number of findings, one gene at a time, there are a number of cases where we've done some things. In fact, we got some pilots going on with Dr. Denny right now around type 2 diabetes. So we know that there are environmental perturbations that can be influencing type 2 diabetes outcomes. We've got a pilot, not with all of the, 700,000 participants, but we've taken a small cross-section of them, and we're studying them in detail now. We're also looking to bring what we call the GIS coordinates. So knowing where someone lives and where someone works, or where they've lived and worked in the past, we can estimate what their air pollution load is and other variables, whether they have contaminations in their drinking water. So that's the first phase of the things that we're doing with Josh and his colleagues at the All of Us program. And then once we've gone beyond that, we'll do this small pilot study where we can go in, take small samples from the individuals, analyze them with what we call high resolution mass spectrometry, where we can go in and we look at potentially thousands of different chemical entities within an individual system. So we're making a lot of progress. In fact, our prediction is that pilot project in type 2 diabetes will increase awareness of the importance of doing gene by environment studies. I don't know, Josh, did I accurately kind of summarize the work that we're doing together on type 2 diabetes? And of course, a lot of this, with the exposome concept, fully integrates the nutrition and diet into the framework of what we're doing.
Wonderfully, Rick. I love the encapsulation of really trying to get the whole picture of health, trying to get all those things, elements that influence it, whether it be, we talk about the genome. I talk about the genome, as Rick points out, a lot. I think a lot about drug-gene interactions as well, which is another aspect of that. But, all of us was designed from the beginning to be able to capture those environmental exposures, to be able to ask questions about social determinants of health, get at sleep data that we get from wearables. And, we're going to start getting from questionnaires as well. And even things, cognitive and behavioral, interactive brain games, so to speak, to get at more information. It's not just your traditional surveys, really getting the whole picture of health. And, the programs like what Holly is leading, building off of a platform, we'll have learnings that we can go apply to the rest of the cohort that we take this deep dive in randomized trials like Holly's talking about, like Rick is talking about, with doing a deep molecular measurement on a subset. And then, maybe we learn something from that we can apply across the whole population. And eventually these technologies will also get cheaper, and we'll actually probably run them just like we do genomes on the whole population as well.
Josh, is epigenetics something that you're also taking into account?
We are, yes. And Rick may want to talk about this as well, because he's more of an epigenetics expert than I am. We are generating one of the world's largest set of what are called long read genomes, which include some epigenetic markers. Rick, do you want to talk some more about that?
Absolutely. And in fact, so just so your audience is aware, what is epigenetics? So we all know about the DNA sequence. There are 4 different bases that comprise the genome. And the genome, of course, is what defines individual biology. But what we've determined, and a lot of this has come from the environmental health sciences community, there are certain environmental exposures that can actually chemically modify some of the bases. The C base in particular, it can methylate it. And when it does that, it can actually change the way the genes are working. So you can change the way a gene works either by mutating it, by changing the base sequence, the code, the genetic code, or you can change this chemical modification of those bases. And that can change the way the genes are working. In fact, work that I'm doing in my own research lab is collaborating with Josh in his own research lab. We're looking at the influence of some of the epigenetic variables in the All of Us cohort. So in the end, what we hope to have is not just the nucleotide or the genetic code sequence, but we could also look at the epigenetic code across the members of the All of Us program. And what we'd want to be able to do is to correlate how both the genetic and the epigenetic changes. And many of those epigenetic changes are going to be influenced by the environmental exposures. In fact, we think that many of the environmental exposures are actually mediating their effect through these epigenetic changes in the genome.
Wow.
I can
fascinating.
go on, but that's, I
No,
think we
so
stop at that point.
yeah, no, thank you. So there are a lot of gaps in health data, and Holly, you talked about electronic health records, which is often, siloed. What are some of the other long-standing gaps in health data that all of us is helping to close or showing ways of closing?
Well, I do want to emphasize the electronic health records because this is not trivial. Nutrition and diet are typically not part of the electronic health records. So we are collecting this data and having it in the same place, which is huge. We're in the middle of a diet-related chronic disease crisis. Yet, this is not something that is typically asked about by healthcare providers. So we will have longitudinal electronic health records data plus nutrition data available to researchers for this purpose. But within the Nutrition for Precision Health program, we're also able to leverage the genomic data. We don't need to collect that and analyze that twice, representing a huge cost savings. And All of Us is an excellent platform for collecting survey data or participant provided information, where it's pretty easy to reach back and ask about social determinants of health, or they very rapidly deployed a survey during COVID to understand everything about how the pandemic was affecting different people. So having this platform to continuously engage participants who are eager to share information about themselves and eager to advance scientific research is the perfect platform for conducting nutrition studies.
Can you just remind me where the genetic information comes from?
Genetic information is collected from participants. When they sign up for the All of Us research study, they have the opportunity to donate a biospecimen, either blood or saliva.
Okay, great, thanks. So this is
Jenny, if I could just add a comment.
Sure.
Just like as Holly was mentioning, the nutrition data is not part of the electronic health record. Actually, for the most part, the environmental exposures is not part of the electronic health record. We want to change that. In fact, my physician has never asked me where I work, okay, or where I live. And what we're discovering is that where you live and where you work actually can predict what some of your environmental exposures are. So it's really combining all of these things and really bringing sophistication to the electronic health records. And Josh referred to, in fact, he might want to expand on this, some of the survey data that they're collecting as part of All of Us. And some of that, well, they're also collecting addresses. So where can you live? So these are all really important things that I think will bring more robustness to our analysis of chronic disease.
And I just want to remind our audience, we're not going to call on people. We don't typically do that. But if you have a question, please put it in the chat or in the Q&A, and we will get to it. Let's see. Got some really good questions here. What kinds of health challenges do you think All of Us is uniquely suited to help tackle? Obviously, environmental exposures, nutrition would be two of those. Are there others, or can you kind of typify, what are the ideal types of questions for All of Us?
In some ways, it's a challenge to answer your question because the number of applications is so broad. And that's our real strength. We are not just focused on cardiovascular disease, cancer, drug exposures, environment. We're kind of focused on all of it because all of it affects health. We will all eventually die from something, and it could be a rare disease, it could be a common disease, it could be some mix, it could be an adverse drug event. All those things are the kinds of things we actually tackle. So, we have hundreds of thousands of people that will have cancer. We have, I think, approaching 100,000 people that have some sort of heart disease. These kinds of are examples that you can take a disease and then you can go look at, the genetics, the nutrition, lifestyles. We can focus to one of Rick's earlier points, because we're getting electronic health records, which goes back in time. We have 40 years of electronic health record data in some people. We can ask the questions that are not part of the medical record. As an aside, Rick, I used to be a primary care doctor. I did ask my people where they worked. My patients, we talked about that part, but I didn't get into the environment exposures. And that's where we can get smarter, because when we learn, as everybody knows now, lead's a problem, then we start asking people if they had, a house that was painted in prior decades. Are they likely to have asbestos in their home? What is their job, and are they exposed to certain kinds of exposure? I grew up in Kentucky, and had family members that worked in Eastern Kentucky. And so, it was an important thing around coal exposure and coal dust exposure and some of the unique illnesses that happen with those. So we can integrate all those things. And we are really, I think, well situated by having a large population, by having a truly national reach, to be able to address a lot of those questions across really all diseases. We have 18,000 researchers using the product. And just about any condition you look for, you're going to find people looking at, whether it be rare disease, common disease. We even have people looking at the old EHR data, even though we don't have many children in yet, the old EHR data from when they were kids, because they share that historical information after they enroll.
Speaking of kids, one of our audience questions is, what's the status of including minors in all of us? And what plans exist to continue that data collection if it's occurring now?
So the plan for all of us was always be across the lifespan. It's really important to collect all ages, and what happens at an early age can certainly have influences on what happens, strong influences on what happens through your life. And we started with adults. We had always planned to develop a pediatric program with a goal of 150,000 children enrolled as part of families in all of us, so that they have a parent or guardian and sometimes multiple other family members as part of the program. We have successfully beta tested with children at 5 sites, enrolling them the earliest ages 0 to 4 in the program, because of decreases we saw in funding due to the 2 sources our funding are 21st Century Cures Act and annual appropriations. Due to decreases in the 21st Century Cures Act in fiscal year 24 and 25, we haven't been able to expand that as we've liked, but we do still hope over time to enroll a large cohort of children that are part of understanding the complete picture of health across the lifespan.
Very good.
If I could just chime in here just for a second, and kind of emphasizing what Josh and his colleagues are doing with all of us is really building a universal platform that can be utilized by all of the institutes and centers across the NIH studying neurological disease, studying cancer, studying kidney disease, studying aging. And we're hoping to bring the environmental parameters, the exposome, overlaying that data on this universal platform that Josh and his colleagues are doing. So this is a way that we can bring the power of the exposome, power of the genome, and bring these powerful tools to study the etiology of human chronic disease. Really getting to, as Secretary Kennedy wants the NIH to do, getting to the root causes of what underlies chronic disease. Why do we have this increasing incidence of type 2 diabetes? Are there things in the environment? Are there genetic contributors? So it's really an awesome platform. So individual institutes and centers don't have to build our own cohorts. Josh has done a remarkable job of building a highly diverse platform with large enough sampling so you can generate statistically significant data. So I really applaud him for his leadership on this program.
If I could add to that as well, not only is it scientifically advantageous to conduct these studies in the All of Us research program, but fiscally, it's really great. We estimate conservatively that our study is saving on the order of tens of millions of dollars by embedding within the All of Us research program. And the savings come from things like working with participants who are already engaged. Recruitment costs in clinical trials are astronomical. We have a backlist of participants who are ready to join the study, saving us months and many dollars. And leveraging that data infrastructure, making it available to these 18,000 researchers across the country, around the world, who are going to continue to make discoveries even after this study has ended, and housing the data and keeping it secure. All of these things really contribute to our cost savings as well.
So we've got some really great questions, and also some that were pre-submitted. So I'm going to go back and forth between them. Let's see. Does all of us provide clinical trial ready data for drug development? And is the program free for patient advocacy groups?
Yes, I can take that. And if you want anyone else wants to chime in, feel free. Researchers can come to all of us. There's not an access fee. Come in and you use our data on our cloud to do research. And that includes people from participant advocacy groups and disease advocacy groups. One of our earliest, most active users when we first launched was from a patient advocacy group. And so we do have researchers in maybe this non-traditional scientist kind of role that are doing really great work on the platform. In terms of launching randomized controlled trials and aiding in drug discovery, one of the, I mentioned earlier that one of the newest classes of cholesterol medicines, and by far the most powerful, came from genetic studies in diverse populations of different genetic ancestries. And we have already seen a couple other discoveries that are leading to how we, for instance, could tackle a genetic cause and actually the major driver of extra risk of chronic kidney disease and need for transplant or dialysis in African Americans, and points to how we could treat that. Holly is leading a randomized controlled trial based out of all of us. It's not using a drug, it's using diet, but that same sort of model could be deployed for doing a drug study that could feed and derive from things like other molecular measures. And we even have a study that will launch soon that's looking at people. Let me tell you, about 2/3 of the genes out there, we don't really know what they do in terms of disease. 2/3. We've figured out the map of the human genome, but we don't know what a lot of the underlying biology is. And some of those are going to cause diseases, and some of those are going to be intervenable. And so we have a study that probably can only be done on our platform, where we're looking at people that have genetic changes that we don't understand that look like they might associate with disease. And then they're going to be contacted for more information and brought in to do studies at the NIH Clinical Center to, maybe define new disease conditions. And potentially that will lead to new treatments.
Thanks, Josh. So interesting. Does all of us have AI applications in your workbench that would help researchers with specific searches? For example, selecting the type of data they need in their own trials.
A great question. When you started to talk about AI, I thought you were talking about, are people using it for AI? And of course they are. Holly gave one example of the glasses with cameras attached to them and learning what people eat. We have a series of tools in our workbench that help guide people in how to use the system and make it easier to do research. We have not built systems directly yet in the workbench that are using AI to help dynamically do things and make it easier for someone. Although we're actually working on a couple systems like that to aid in writing code for deeper analyses and things like that. But lots of people are using it for AI discovery now, because the first thing you need is that data set. You need a clean data set that's got great representation across like different EHR systems, different parts of the country, different exposures, all those kinds of things.
Yeah, if I could just chime in here, I think the AI the timing is perfect. So, Josh is bringing in these awesome data sets around the genetics of up to 1 million people. Holly is bringing in these phenomenal capabilities around nutrition and changes. These are all huge data sets. And in the past, we've looked at, one thing at a time. What's the influence of genetics on disease outcome, or diet, or specific environmental exposures? With AI, we now have the ability to integrate across all of these things and to better understand how biology actually works. And it's not just genes, or it's not just diet, it's not just the environment. So that's where I know that there is a lot of interest, certainly at NIEHS, our grantees, building the tools to integrate across all of these complex data sets.
So one of our questions is, what's the potential funding situation with all of us? And will there be additional participant recruitment in the upcoming year And I know that from where you each sit at NIH, you're not in a position to discuss the budget, but I certainly can from Research America's perspective. The president's FY26 request included just the 31 million from the Cures funding. And as we all know, there was quite a large cut, 40% in the president's budget. Research America is very actively working to address that and working to, for at least level funding, if not more, across NIH. And I think there was also a dip between FY23 and 24 in all of us. But the funding base in 24 and 25 was about 122 million, and then with additional Cures funding. So that's the funding picture. I don't know if, Josh, you want to answer the part about additional participant recruitment, or is that a wait and see?
Yeah, I guess as we've seen a change in our funding due to the decrease in Cures funding, we have had to prioritize. And that's been around our current participants and support for researchers, support for these as a platform for the kind of research studies that you're hearing about today. That's meant that we've had to slow and almost pause a lot of our new enrollment. Because we've created a digital platform, people can still come through the front door. We just don't have the funding to collect the biospecimens at the same rate of people who want to give them to us and generate the genomes at the same rate that we could in prior with the funding we had in the past.
Mm-hmm, mm-hmm. Great. Let's see. Question. Is there any interest or thought about engaging with international partners or data sets, especially those with national healthcare systems that have longitudinal health data?
Definitely. We actually helped start something called the International 100,000 Person Cohort Consortium. IHC, it's a mouthful, but it is all these groups across the world that have biobanks and of different ilks and working together, trying to create national standards. There's another group out there around genomic standards that we're heavily involved with in how we create, and we've actually had to, given the size of our data, we've had to create and invent basically new approaches to how we store and manage the data sets of these sizes, especially around genomic data. We've really been pushing on what the standards are around phenotype data representation. So the genotypes are actually really easy to standardize. The phenotypes are really hard. So we've been pushing on that. And we really do routinely work across the world in terms of how do we harmonize and get these data sets, so these cohorts, so that people can take them and make it easier to combine those data to create larger data sets for discovery.
Yeah, I can also add,
please, Rick.
A lot of interest in the global community to do exactly what Josh described for the genetics and genomics communities, doing it with the exposomics communities. Europe is quite a bit ahead of us in terms of developing cross-nation collaborations and develop standardization for data sets. The United States is now entering into that, but we want to be able to do the same thing. So when we're collecting the exposome data and when we're creating those epigenetic profiles, we want to be able to standardize it in a way that there can be seamless of sharing of data across the geographic boundaries.
So let's jump from the research to the application. And, there are a lot of challenges in access and delivery of healthcare in the US. How can your findings help to relieve some of this? Is there a way that the data and information that you're gathering and then really testing, how can that help? How can that be delivered to folks in the healthcare system? Clinicians, hospital administrators, et cetera.
Well, maybe I'll give a perspective as a practicing primary care physician first, and I would love for others to chime in. I did give up practicing in 2020 to be fully devoted to this project. But, we asked questions of those things that we knew how to intervene on in medicine. and that we knew, and I gave examples of lead and asbestos. We rapidly adopted new technologies when we knew, like CT scans and echocardiograms, when we knew we had predictive value and could make someone's care better to then guide what their therapy is or prevention. And so part of it's the simple act of discovery and getting the information out there. When we find a new treatment and that treatment knocks the socks off the old treatment, and we can target a therapy and really only do the colonoscopies, the dreaded colonoscopies, on the people who really need them, we will adopt them rapidly. The second thing to talk about is, we can get smarter with how we build healthcare systems and embed some of these informations into the electronic health record as we know that. That's something Holly was talking about earlier about diet information. Well, if we get smarter about, from this program, how we can define from the diet-ome, if we can use that word, to the specific kinds of questions we want to ask. And then the last thing, I just want to highlight that one of the things we do is return genetic information to participants that want it. I always had this vision that returning pharmacogenetic information, what drugs and genes interact, is one that everyone should want to have, because it's just about taking the right medicine to work the first time without side effects, with the best outcome as possible. And a lot of times that saves you money. I used to do this in my practice. And that means you could use the old cholesterol statin medication that was generic in some people and push the dose, and they're never going to have a side effect. And then in others, maybe you need to tailor that to avoid a side effects. So it can save money. It can save money, deliver better outcomes. And by returning this to hundreds of thousands of people, we're getting doctors used to seeing this kind of information across the country. And hopefully that will help lead to greater adoption of genetic medicine as one tool that will, benefit everybody.
Thanks, Josh. Oh, please, Holly.
I'd love to expand from the nutrition side. So right now, if you go see your provider, if you see your registered dietitian and you're looking for dietary guidance, they're going to ask you some of the usual things: your age, your activity level, your goals, and so on. But to expand on what Josh said, knowing what to ask. Right now, they're not really asking about the microbiome. We're going to learn more and more things that your provider needs to know. And one thing I didn't mention that I think people tend to be interested in: do you really want to go to the doctor and provide a stool sample to get your microbiome tested? But as we learn more and more about this, we can explore ways to make this less and less burdensome for the participants. So one of the things we're piloting in NPH is actually a smart toilet tissue collection device. So all you have to do is deposit your toilet tissue, do everything normally, and it's going to collect it and save it for you until it's ready to be shipped off for analysis. So no extra work on your end. But the ultimate goal is really figuring out what are those things that are going to work for you the fastest. To use an example that I think most people can relate to, why would you try this diet and then this diet and then this diet, each for months at a time, if you're trying to lose weight, for example? When if we can just get some more information, a few more questions that your provider asks, or a few more lab tests, then we know this one is going to be right for you. Skip all the drama and the heartache and go right to that one that works.
This is so exciting. We could keep going, and unfortunately, we're at time. Before we end, two things. One, if potential users want to review data that has been published, is that on your website? And then also, how can researchers contact you? Is that also on your website?
Yes, to both of those questions. Researchallofus.org has information about that. You can look at the publications that are coming out. We have a backlog of putting them on the main page, so we've put another page that just kind of gets a quicker list of what they are as they're coming out. We're getting up to 20 applications or publications a week now, and then in terms of reaching out to us, our NIH emails are all pretty easy at this point. It's our full name our last name at nih.gov. But if you look at our bios, I think our emails are probably each in there.
Well, Josh, Holly, and Rick, I'm going to make some closing comments about our next meeting, but I can't thank you enough. This is so fascinating, and we really applaud what you're doing. So thank you so much for joining us today.
Thank you.
And for those who are members of our alliance, we are going to have a call next Tuesday, the 15th at noon. You'll be receiving information about it, but we'll be talking to Cheryl Yeager of Crossroads Strategies about the current policy and funding landscape, including but not limited to appropriations, reconciliation, and the possibility of funding rescissions, i.e. federal funding that is returned to the federal treasury. So as always, bring your insights and your questions to next week's meeting. And if you're an Alliance member, you can email Jacqueline Lagoy. Her email's in the chat for the Zoom registration link. So thank you all for joining us. What a great crowd. We had almost 200 folks who stayed on the whole time. And we thank our friends from NIH.